Welcome back. This is the third part of our series on the central control of reproduction. I've entitled this one, Opening Up the Black Box, Negative and Positive Feedback. Why opening up the black box? Well, this will become apparent, but essentially we're seeing the brain, especially the hypothalamic part of the brain, as a black box. And we're going to be investigating the inputs into it and looking at the outputs from it and see if we can interpret, because of those things, about what's going on inside. So this is our black box, really the hypothalamic area of the brain with many inputs into it. We know that the GnRH neurons live there and they're somehow coordinated together as a GnRH pulse generator system. The GnRH comes out, controls the anterior pituitary secretion of LH and FSH, and one of the fundamental features of that is that LH comes out in pulses. Now, because of this, we can look at the inputs into the system, because there are many of those, and those we can manipulate. We can monitor the outputs either through LH pulses or through what's going on in the ovary and the testis. And by those mechanisms, we're going to try and look at what's going on inside this whole area, how this whole system works. We're going to be concentrating on this, the operating characteristics of the drive to the gonads. Really the LH drive to the gonads, because that's what we can easily monitor, but that's being a reflection of the GnRH drive itself to the anterior pituitary. And the first thing we'll concentrate on, just briefly, is that the system is suppressed in both sexes by negative feedback of gonadal steroids. These gonadal steroids may work both at the pituitary and at the hypothalamic level, but it will be at the hypothalamic level that we'll be looking. So negative feedback at the hypothalamus. Here's an example of that negative feedback operating. We're looking at a rat and we're looking at the administration to that rat of estradiol. So here we have the pulsatile secretion we see normally, and if we give the vehicle, and the vehicle, what that means is the solution in which estradiol is going to be given. Here's the vehicle. In fact, giving the vehicle makes little difference to the frequency of these GnRH pulses. On this one, let's look at the LH levels when we give estradiol itself. Estradiol, the number of LH pulses goes down and the overall levels of LH consequently go down as well. So that illustrates negative feedback of estradiol. We'll come back to the negative feedback system towards the end of the lecture, because we're going to concentrate for most of the lecture on an entirely different system, a positive feedback response, something which generates the pre-ovulatory LH surge, the LH surge which causes the follicle to rupture. And we'll see in a minute why it's called positive feedback, but it's partly to actually contrast it with the system we've just looked at, where estradiol normally induces negative feedback. Here we see some typical hormone levels as we go through the menstrual cycles of a woman. What we have up here is as the follicle grows, the estradiol levels rise rapidly and reach a peak about mid-cycle. After mid-cycle, after ovulation, progesterone levels rise because of the corpus luteum. And at mid-cycle, the stimulus for ovulation itself is this big surge of LH. Now, why should there be a big surge of LH? Well, what happens is that estradiol, the rising levels of estradiol, if they're high enough and maintained long enough, generate feedback which produces this LH surge. Estradiol-induced LH surge, the cause of ovulation. Here we see in a little bit more detail exactly the same thing in a sheep, in a ewe. This is looking now in terms of hours. Here's the LH surge, which is going to cause ovulation. What causes that? It's this rising levels of estradiol coming from the growing follicle. And they generate the LH surge. How? By stimulating GnRH. So this top graph shows GnRH levels in the portal blood vessels. So we can see rising levels of estradiol initially induce negative feedback, but then that switches to this big surge of GnRH, the so-called positive feedback. And one more example. This is in the rat now, just very diagrammatic. Here we have a time base along here where each little section represents 1 day. So if we give estradiol to an over-optimized rat, suitably hormone treated, give estradiol, that induces an LH surge. That would, in a normal animal, induce ovulation. But an interesting thing is males cannot show the same response, or do not show the same response. Here we have males given the estradiol to try and induce an LH surge, but no, they don't respond. So there's something different between females and males. Females show this estradiol-induced LH surge, males don't. It's a sexually differentiated response, which is very marked in rodents. We're now going to look at how that sexually differentiated response can come about. Here's a mother rat who gives birth after 21 days of pregnancy. Here's one of the newborn males she gives birth to. We allow him to grow up, and we're going to ask the question, can he show a estradiol-induced LH surge when he's adult? And we've already seen the answer, and the fact is, no, he can't. Right. So let's go on to the second experiment. And the second experiment involves a mother giving birth, but then very early on after birth, the newborn male is castrated, his testes are removed at birth, let him grow up, and then yes, he does. He shows the positive feedback response to estradiol, a typical female response which is lacking in a normal male. So just by removing his testes at birth, he develops a female response. Well, why could that be? Well, the obvious thing is the testes, we know, are important endocrine organs. They secrete testosterone, so we can give testosterone back. So let's try that. Third experiment, remove his testes at birth and then give testosterone, and we can give it back at any particular time. But experimentally, the thing which works is giving testosterone back in the first 1 to 5 days postnatally. Then let the animal grow up. He doesn't then show the positive feedback response to estradiol. We've changed his system back to a normal male. So what we've got is a critical period early on in life where if the male rat sees testosterone, then he doesn't develop the positive feedback response. If he doesn't see testosterone, just because you've removed the testes and not replaced them with anything else, he does show the positive feedback response. So it's a sexually differentiated response determined by testosterone exposure very early on in life. And it's a critical period. You have to do it within this first 1 to 5 days of life. So there's a clear take-home message from that series of experiments, which is that brief exposure to hormones during sensitive periods early on in life can permanently affect physiology, and we'll see at another time that it can affect behavior as well. So critical sensitive periods early on in life when correct or incorrect hormonal exposure can change things permanently. That sexual differentiation in estradiol positive feedback is also reflected in just looking at the structure of the brain between males and females. This is a cross-section of the hypothalamus of a male rat. This is a cross-section of the hypothalamus of a female rat. The sexually dimorphic nucleus of the preoptic area is here. And it's called sexually dimorphic nucleus. Why? Because it's different between males and females. And this is an area which we believe to be associated with the control of that estradiol-induced LH surge. Interestingly, we can look at this is a male, this is an intact female. What happens if we give a female testosterone early on in life? There it is. Female treated with testosterone neonatally, we essentially make her brain structure just like that of a male. So female with testosterone neonatally, and she can't show the estradiol-induced LH surge. So a sexually differentiated response due to testosterone exposure early on in life. not only a physiological difference in terms of the estradiol-induced LH surge, but an anatomical difference up in the hypothalamic area of the brain as well. Let's go back to the black box model, which is the brain, the outputs being the pulsatile secretion of LH. Here the GnRH neurons coordinated together somehow with a GnRH pulse generator system. Many inputs into the system. Negative feedback of gonadal steroids works by suppressing the GnRH neurons and suppressing GnRH pulse frequency. But we've also seen with a good model in the rat of a positive feedback effect of steroids, which is sexually differentiated. Apparent in females, not apparent in males. But let's now add one other factor into this, another input. What could it be? Time. Time has a distinct and specific gating effect on this GnRH pulse generator and the positive feedback effect of steroids. Let's look at what we mean by this, the time. Well, rats tend to mate at night. So here's a male mating with a female, and he tends to do it in the middle of the night. Why? Because if he delivers his sperm there, that's about the time when ovulation is going to occur. If we take some controlled rats on very specific lighting, the lights come on at 6 o'clock in the morning and they go off at 6 o'clock at night. So all this period is dark. Ovulation in these rats would occur about 2 o'clock in the morning. Now, why should it occur at 2 o'clock in the morning? Well, ovulation is induced by LH and a big LH surge, and it takes about 12 hours in rats for that ovulation to be triggered by the LH surge. So the LH surge occurred 12 hours earlier. That is about 2 o'clock in the afternoon. So there's obviously a time of day signal somehow involved in the LH surge. If it occurs at a specific time of day, why is that? Well, we know that estradiol induces the positive feedback, the LH surge. So high levels of estradiol, if we give it estradiol here, they will induce the LH surge, and it occurs about 2 o'clock, and then it goes away. But the peculiar thing is that even if you keep the estradiol levels high all the time, the LH surge goes away, it goes away, it goes away, and about 2 o'clock the next day, there's another LH surge, and then it goes away again. So even in the presence of maintained estradiol levels, maintained high estradiol levels, there's a daily signal for the LH surge on top. It isn't just the estradiol that's important, there's a time of day signal as well. So it occurs on day 1, day 2, day 3, and onwards. And it will go on for about 10 consecutive days. Maintained estradiol levels, a daily LH surge. A daily signal to the LH surge. What's going on? Well, we'll come back to how the time of day and the timing system actually interrelates with the reproductive system in a later part of this whole series. But up to now, we can conclude this. The pre-ovulatory LH surge in rats depends on estradiol positive feedback. There's a sexual differentiation of the brain, so it occurs in females, it doesn't occur in males. And there's an input somehow from the daily circadian system. We're now going to use that estradiol positive feedback system, which occurs in the female rat, to try and open up this black box of the brain, how the estradiol, that's the input, interacts with the output, the GnRH neurons which generate the LH surge. So this is the positive feedback loop, estradiol somehow connecting up with the GnRH neuron, inducing the LH surge. Now, obviously the first easy hypothesis would be this, that high levels of estradiol somehow act directly on those GnRH neurons to induce that LH surge. But that has a problem, because these GnRH neurons don't have any estrogen receptors. There's more than one type of estrogen receptor, but the main one is estrogen receptor alpha, ER alpha, and GnRH neurons don't have any estrogen receptors. So if they don't have any estrogen receptors, this hypothesis can't be true. Estradiol can't be acting directly on the GnRH neurons by the traditional receptor-mediated route. So what would be the next hypothesis? The next hypothesis would be, okay, there must be other things feeding onto these GnRH neurons which perhaps do have estrogen receptors. And certainly we know that there are a lot of synaptic inputs into the GnRH neurons, and we know that some of those certainly have estrogen receptors. So that's our next hypothesis. A new player entered the field in the early part of the 21st century. Right at the turn of the century, which was an era of molecular biology and discovering new receptors and new ligands, and often things were discovered without actually knowing what they did at all. And just at the turn of the century, a new G-coupled receptor was found, they called it GPR54, and they also discovered a ligand for it called kispeptin, but nobody knew what these two did. Then in 2003, a very interesting observation came up. There were humans which were infertile, and they seemed to be infertile with an association with mutations of the receptor of GPR54. And in the same year, when people started playing around with knocking out particular genes in mice to see what they would do, if you knocked out GPR54 in mice, or if you knocked out kispeptin gene in mice, they became infertile. So maybe the kispeptin GPR54 signaling played some major role in controlling the whole of the reproductive system, perhaps in controlling GnRH. And that seemed to fit. There was extensive studies of the role of kispeptin in controlling GnRH neurons, a whole series of very logical investigations dissecting away the system. So there was this: high levels of estradiol upregulated kispeptin expression in the neurons in the hypothalamus. Those kispeptin neurons would project, and did project, to the GnRH neurons. They had terminals onto the GnRH neurons. The GnRH neurons had kispeptin receptors, the GPR54 receptors. If you administered kispeptin to the GnRH neurons, that would induce an LH surge. And kispeptin would activate neuronal activity, electrical activity in those GnRH neurons. And one final thing, kispeptin antiserum, if you administered that into the area where the GnRH neurons were, kispeptin antiserum, so you're essentially blocking kispeptin action, it would block the LH surge. Cutting a very long story short, the kind of picture that developed was something like this. Here the GnRH terminals running back to the cell body, lots of inputs onto this axon here, onto the cell body itself. So here's the GnRH neuron cell body. Onto this, kispeptin neurons would feed. Kispeptin neurons are found in the preoptic area. Those preoptic area kispeptin neurons were sensitive to steroidal feedback, but they also had the ability to have many other inputs into them from all kinds of different sources, including time of day. So here we have a time base along here, and here's our preovulatory LH surge. So we have steroid feedback onto the kispeptin neurons, time of day input somehow onto the kispeptin neurons. And one final thing, these preoptic kispeptin neurons seem to be essentially sexually differentiated. There are far fewer of them in males than there were in females. So a sexually differentiated area of the hypothalamus controlling GnRH, which is just what we want for explaining the sexual differentiation of the preovulatory LH surge. But what about negative feedback? Well, that seems to be explained probably by the kispeptin neurons which exist in another area of the hypothalamus, the arcuate nucleus. So kispeptin neurons exist in the preoptic area and the arcuate nucleus, and these arcuate neurons seem to be the primary site of the pulsatile secretion of LH that you get for most of the time. not the pre-ovulatory big LH surge, but just the hourly or 2 hourly or whatever, however frequently it is, the pulsatile secretion of LH. And this may well be where the negative feedback really centers. So positive feedback up here on the preoptic area neurons, negative feedback working somehow on the primary control of the pulsatile secretion. These kisspeptin neurons often express also neurokinin B and dynorphin, so they're called KNDY neurons or CANDY neurons, and you'll learn more about those if you read more about the GnRH pulse generator itself. Well, that's it for this part of the lecture. Thank you very much for listening.